Hello, my name is Anya, and I'm learning single-cell RNA-seq analysis. This channel is to motivate myself and to share knowledge with others. In my learning journey, I use publicly available data and published paper. For example, today I'm using this paper, which looks into the effect of chemotherapy on tumor-associated microenvironment in pancreatic ductal adenocarcinoma. And I will hopefully walk you through how to use AddModuleScore to generate a figure that looks kind of like this. It is way too often during single-cell data analysis when we want to classify the cells based on their expression of a known set of genes. Here we have a UMAP. This UMAP shows the cancer cells in pancreatic ductal adenocarcinoma grouped by the sample and the patient. And also we have here a set of genes that classify the cancer cells into classical and basal. These are known genes that were previously described in this paper. Here is how to use AddModuleScore to classify each cell into basal and classical. Before we start with the example, I need to make two disclaimers. In the reference paper, they use samples from two sites, liver and the primary site. Here, I use only the samples from the primary site. And also, you saw here in the UMAP that the samples are separated by their basically samples. The cells are separated by their samples. It looks like the data is not well integrated. However, this is an integrated data, and this is how it looks like in the original paper. This was one of the findings that the pancreatic ductal adenocarcinoma cells are very heterogeneous, so it looks different for every patient. Now I will show you an example of how to use AddModuleScore to classify the pancreatic ductal adenocarcinoma cancer cells into basal and classical based on a known set of genes. Here I have a Seurat object that has all the cells, including the cancer cells and the tumor-associated microenvironment cells, including the lymphocytes, like the myeloid cells, the cancer-associated fibroblasts. And this Seurat object has been pre-processed. So I've already done the quality control, the doublet finder, and the integration. Now I will show you an example of how to use AddModuleScore to classify the pancreatic ductal adenocarcinoma cancer cells into basal and classical. I will need to make a new Seurat object that has only the epithelial cells. So I've already run these steps. Basically, I will subset the epithelial cells in a separated Seurat object, and then I will perform the data normalization, scaling, and the dimensionality reduction and reclustering to, again, on the new subset. So here I want to show you how this subset looks like on a UMAP. Yes, so as you can see, these are only the epithelial cells and grouped by the samples, and you can name it the sample identity. Because pancreatic ductal adenocarcinoma is a very heterogeneous disease, each patients look different on the UMAP. Now to the most interesting part, which is basically using the set of genes that we have to classify each cell into each subtype. Here we have a table that includes 25 genes that are specific to classical and 25 genes that are specific to basal. This is the AddModuleScore. The AddModuleScore requires a Seurat object. This is the pre-processed Seurat object that we've just made, and it needs a list of genes basically assigned to each subtype. And this parameter right here tells the Seurat to compare each set of genes against 100 reference genes. These reference genes are not selected randomly. These reference genes are chosen from the same expression level as the genes of interest, and I will link this article here that explain how AddModuleScore works internally. And finally, the names that we will be assigning each subtype to. So now if we go to the metadata of the new Seurat object, we can see that there are two columns, classical and basal, and these are the scores. So basically, what we need to do now is simply classical score minus the basal score. And if the score is positive, it's classical. And if it's negative, it's basal. This is the AddModuleScore. The AddModuleScore requires a Seurat object. This is the pre-processed Seurat object that we've just made. And it needs a list of genes basically assigned to each subtype. And this parameter right here, we will create a new column in the metadata called subtype. And this column basically tells the Seurat object to call the cell classical if it has a classical minor basals above 0, and if it's below 0, it's basal. So it's basically common sense. Let's look at the metadata here. This is the subtypes, and this is the classical minor basal score. Now, let's show you how this looks like on a UMAP. As you can see, we successfully classified each cell into classical and basal based on a known set of genes. I hope you found this useful, and thank you so much for watching.